this is one that is useful, and over the years it has become very popular. And now these standards vectors, these standard systems that I want to tell you have become a very good tool for engineering a large variety of microorganisms, not only E. coli, but also other bugs of interest. Okay, so standard European vector architecture. So what do we mean by that? I'm sorry that we have this kind of chauvinistic name here, European, but we put it there just because it sounded more euphonic, you know, the SIVA sounded good. Okay, so this idea started 10 years ago, and the idea is the following. Until that time, plasmids vectors had an entirely, say, disorganized architecture. So in a plasmid vector, you need to have some functional modules. You have to have an original replication. You need to have a antibiotic resistance marker. You need to have a place for cloning things. But the thing is that different laboratories had very different configurations of different plasmids. And on top of that, most of the plasmids by that time were developed exclusively or nearly exclusively for use in E. coli. Okay, that is like the workhorse of many laboratories. Okay, then at some point, say, why don't we make an effort and try to standardize plasmid vectors following very single rules? Okay, and this is something that ended up with this initiative that is called the Standard European Vector Architecture. So the Standard European Architecture that is now in its 4.0 stage is about having plasmids engineered, vectors engineered in a modular fashion in such a way that the result has many advantages in respect to what we had before. So I have this video here that will give you a snapshot. No, wait. Can we connect the link here? Can we click here and see what happens?
We have learned that the best way to tackle complex problems and make things work in lab science.
Yeah. Okay. All right. Thank you. Thank you very much. All right. So what is the standard about? The standard is the following. Let's imagine a plasmid, and then in this plasmid, we are going to have it like a circle. And then in the circle, we just establish 3 fixed components. And the components are exclusively 2 terminators, 2 transcriptional terminators, and an origin of transfer. An origin of transfer is something that enables these plasmids to be mobilized through conjugation from one host to the other. And this is important because you may know that some bacteria, some microorganisms are very difficult to transform through chemical means. And the way to make them get external DNA is through conjugation, okay? So any plasmid that has this ORIT, that is a sequence of DNA, a small sequence of DNA, can be mobilized from one host to the other through conjugation. But this is an abstraction, right? You just have a terminator here, a terminator there, and a terminator there. Now, what are the functional components of a plasmid? Okay, then you have an origin of replication, then you have an antibiotic resistance marker, and then you have what you call a cargo, that is actually the part of the plasmid that does something interesting. So these are the three parts. Now, what are, under this general frame, the general characteristics of the plasmid? Okay, well, first of all, is that to follow the standard, you have to make sure that the functional segments that you put in the plasmid or in the vector are the smallest. They are minimized. For instance, if you have an origin of replication from this plasmid that is called RSF1010, it's a famous plasmid because it's super promiscuous, then and the natural sequence is 5.4 kilobases. What you do is that you look at the sequence and try to eliminate from that sequence all segments of DNA which are not essential. And in this case, for instance, we ended up with an origin of replication that was, 33% reduced in respect to the other. Another characteristic is that we make sure that the sequences that we've put in the plasmid have been curated from many restriction sites that are commonly used for cloning. So for instance, if one of these segments have an EQR1, above one site, then the standardized segment that goes into the plasmid has been curated, has been eliminated for that. It is possible because all segments have been synthesized, okay? So it's not that they have been mutated, it's just the segments are synthesized. So one of the components can be this one, but this is a natural sequence, but we have these two sites there that we don't want. So we resynthesize, we edit the sequence to synthesize, to eliminate these two sites. Now, what are the characteristics of the backbone? Well, what I just told you, you have just two terminators and you have an origin of transfer that comes from a natural plasmid that is called RP4. So the total length of the backbone, that is just these three segments, is about 0.5 kilobases. That is very little. And everything else is synthesized. And what is important for the rest of the concept is that excepting these fixed coordinates here, and there, all the others may have different sequences, but these three are always the same in all vectors of the collection. And this is something that has good consequences. And it has consequences because these points in the coordinates of the plasmids are flanked by restriction sites that are very unusual, and therefore they are unlikely to be used for cloning purposes. So we have some of them, like PAC1, SC1, SP1, and so on, and they are very unusual. So the three fixed sites that you have in the plasmid, that are one terminator, another terminator, and an ORIT, they are all flanked by these unusual restriction enzymes, okay? They are rare enzymes now. So the advantage of doing that is that by keeping in all plasmids all these sites, then we can easily exchange different functional segments among different plasmids, and it's really, really easy. On top of that, we develop or we propose a given nomenclature. If you have used plasmids in your daily life, again, the names of plasmids can be completely, say, chaotic. You can give the name of a plasmid in an arbitrary way because of the name of the laboratory or because of the name of your dog, and it doesn't matter. You can give whatever name you want. And this is sometimes confusing, so we propose to have a standard nomenclature for a number in the plasmids in which you have a numerical code, and in each position of the code, you have a number that indicates the antibiotic resistance of the plasmid, the replication origin of the plasmid, the cargo, and the gadget. So gadget is something that you add to the frame to incorporate a new functionality that might be of interest, as you will see a case later. For instance, Well, you have a collection of origins of replication, you have a collection of cargos, you have a collection of antibiotic resistances. So you have, for instance, a plasmid that is called PSEBA 6913 alpha. That means that you have number 6 original replication, that you have number 9 a cargo, that you have 13. Wait a minute. So this is no. Wait a minute. So that you have in this first position, that is antibiotic resistance, you have gentamicin. The original replication is this one that comes from environmental isolate. The cargo is a promoter, and then the gadget, for instance, is a small sequence called HOXOC, that is a kind of trick that we use to stabilize plasmids in the absence of selection. So, so instead of having a kind of arbitrary name, if you have a number, that is 6913 alpha, you know instantly what is in that plasmid. Okay, so that's something that is quite good. Also, because of the conservation of the restriction sites that flank each of the component of the functional components of the architecture of each vector, then it's very difficult to exchange functional modules. So, for instance, you have a cargo for expression of heterologous proteins that is induced by benzoid here. For instance, this one that depends on a transcriptional factor called silase and a cognate promoter that is called PM. But your initial plasmid has a resistance to kanamycin, and for whatever reason you don't like kanamycin because it's really bad, then you can excise that segment and replace it by another segment from another plasmid that has a different antibiotic resistance or a different original replication. I mean, you have a complete modularization, and therefore, you know, you can make all the times different variants of the same architecture to fit your specific engineering purposes. Then also the plasmids are engineered, are kind of the device for easy sequencing of the inserts. Well, I have to say that this idea was developed before DNA sequence was so easy. And even though these days you can just you know, synthesize your, I mean, make your construct and make it have it sequence for just a few euros or a few rupees. By that time, it was more difficult, and therefore we had these primers that would amplify the cargo regions exactly for enabling and facilitating sequencing. Do you have this service for rapid plasmid sequencing that, you know, costs you nothing, basically? Well, I don't know whether, well, I understand that you are still undergrad, so may not be exposed to actual, or many of you may not be exposed to actual experimentation, but right now there are companies that, you know, will sequence your plasmid, I don't know, for a few, maybe 5 euros or something like that. I mean, something that is incredible. You know? Well, in any case, so if you don't want to go through the sequencing, you can always use these sites for primer amplification, for amplification of segments through PCR, and things go much better. Okay, let me discuss a bit one very important part of the plasmid, of the concept, that are the origins of replication. So the origin of replication is, of course, the part of the plasmid that enables the propagation of this plasmid in a given host. Many, I would say the immense majority of the plasmids that you find in the biological literature are plasmids that are intended to replicate in E. coli. But as you know, E. coli is not the end of the world, is not the ultimate host, is not the ultimate chassis. And in the last few years, more and more chassis have been proposed as good for synthetic biology purposes. And therefore, in the collections, we have incorporated a number of origins of replication that enable the propagation of these plasmids, not only in E. coli, but also in a wide variety of bacteria, both gram positives and gram negatives. So the ones that you have on top of here, so are plasmids that are promiscuous, origins of replication that are promiscuous, and therefore SEBA derivatives that carry these origins of replication can replicate in many types of different gram-negative bacteria. And also, depending on the origin of replication, you have plasmids that have a high copy number and others that have very low copy number, both for promiscuous plasmids and also for E. coli plasmids. And this is very important also in some biotechnological projects, because you want to control sometimes the degree of the level of replication of your plasmid, and sometimes you want one that is low copy. In other cases, you want it higher copy. The lowest of all is this plasmid called RK2. So RK2 is, in fact, one of the best plasmids for any type of purposes, because the number of copies is very low. In fact, we consider it to be nearly 1, even though it says 5 here. And because when you have just an extra gene or an extra device incorporated into a pre-existing genetic network in monocopy, then the disruption that you create in the rest of the network is far less than when you add something in multi-copy. That multi-copy plasmids start, sucking resources, and cells are generally unhappy. For instance, you have this POC plasmid. That is a plasmid that is very popular for cloning, but you may have between 500 and 700 copies of the vector, of the plasmid. And the burden that you create to the cells when you have something cloned in POC is tremendous, okay? So if you have a plasmid clone in a low copy number plasmid, then chances are that it will remain stable for a while. But you have something cloned in a very high copy number plasmid, things can get a bit nasty, because the cells will try to get rid of it and will mutate it and will create havoc. There's one a specific type of origin of replication that is quite interesting, and I will return to a bit later, and is the origin of replication of vector R6K. So this type of replication is the interest of it is that the origin of replication depends on a protein that is called the pi protein that can be delivered in trans, and therefore only specialized hosts that produce this pi protein can host this type of plasmid. This is something that is important for some applications that I will tell you later. And we call it a suicide plasmid, because when it can be transferred to a new host. But if the new host does not have the P protein, and this is the case in most cases, then this plasmid will get into the new host, but will not replicate there, will stay there for a while, and will eventually be lost because it cannot replicate. Also, antibiotic resistances here. What antibiotic resistances we can use? In the immense majority of vectors that are used in molecular biology laboratory, the one and only antibiotic resistance use is 1, 2, 3. No. Ampicillin. Okay. Well, it's funny because you tell me exactly the ones that are less used, but anyway. Okay, so most plasmids around are ampicillin resistant, okay? This has a historical origin because the first vectors that were created at the beginning of the era of recombinant DNA happened to have antibiotic resistance to ampicillin. But there are many other antibiotics that can be used, and you have a bunch of them here, kanamycin, chloramphenicol, and so. It is true that in other bacteria, non-E. coli, other antibiotic resistances are used often, but ampicillin remains one that is very popular. And what we did for the sake of, has been done for the sake of the SIVA collection was to take the genes, and I said before, you synthesize them, eliminating some restriction sites and so on, just to make them to fulfill the standard. Then we have cargos. Okay, so what cargos we can have there? Well, the cargos, one would say aspect of the design is that they are flanked by terminators. So terminators, in principle, stop transcription going either from upstream or coming from downstream, and prevent also the the transcription that can be originated in the cargo itself to propagate into the rest of the plasmid. Okay, so I have one thing that is important to know is that even though we call these sequences terminators, I mean, terminators is a very strong word, terminator, like in the movie Terminator, you terminate here, period, and you go. Well, terminators in bacteria are never 100%, okay? So these terminators here terminate to an extent, but I would call them more like attenuators, okay? So you have some transcription going into the cargo or from one side or the other, then these terminators will attenuate the effect of this flanking transcription, but we will not eliminate it completely. But in any case, a part of the cargo, we maintain here two fixed restriction enzymes, PAC1 and SP1, that we can use it for different purposes to confer functionality to the plasmid at the end. Okay, so you have plasmids and we have a collection of them. What we can do with them as genetic engineers? Okay, what do genetic engineers do all the time? Cloning, right? Okay, so then one of the favorite cargos in this type of vectors are sites for restriction cloning and also for cloning segments of DNA through other methods. So, well, you have a typical polylinker here. You can use restriction cloning with many enzymes and so on. And therefore, I mean, of course, these plasmids can be used for cloning in general. Some variants of these polylinkers, of these multiple cloning sites, are also combined with this trick with the alpha laxed segments so that you can discriminate inserts through the white blue screening method. I don't know, you are all of you familiar with them, but you know, this is a method that enables you to discriminate visually those clones that have received an insert from those clones where you have just relegated the plasmid. Okay, you can also quantify gene expression, and this is something that is very nice in many respects, because quantification of gene expression in general is made with reporters, and reporter genes are kind of fun to use because they deliver nice output signals. So in that case, what we have is a number of plasmids that are promoter-less, so we can enter a reporter, are promoter-less, and we clone that in front of a reporter that will be some output, okay? And obviously the reporter gene will not be present in the organism under study, and then you have some of them that give you signals that are very easy to identify. Laxed is a very popular, or was a very popular enzymatic signal, because you have many substrates of laxed that will give you some color, some blue color, other colors, but others are here, like the lux operon, that will give you luminescence. That is also very nice to see. And also, of course, the whole plethora of GFP and GFP variants that are available out there. So laxed is a historical reporter. It is not used these days a lot, but the advantage of it is that it is practically the only reporter that is easy to measure quantitatively and in absolute terms. So someone years ago decades ago, proposed to have these units of beta-galactosidase, and to this day, you know that you have a promoter with 10 units of beta-galactosidase. You know that it's a weak promoter, and it has 20,000, it's a strong promoter. So you have an idea of what the strength of a promoter is, okay? And the advantage also is that there are many different types of substrates for laxed that have a growing degree of, say, sensitivity. So some substrates like XGAL will give you this blue color, but others will give you a fluorescence, for instance, or luminescence, and also will give you some black color. I mean, you can really use many different substrates that have been organized during the history of the field that make laxed a very appealing reporter. Then you can have, for instance, a reporter lux light, light that can be visible with the naked eye, or if not, with a luminometer. And in that case, what you have is this enzyme that is a luciferase that is formed by expression of all this bunch of genes here. And then you have the right promoter in front of these genes here. What you have is emission of light that you can see with your naked eye. So well, in that case, what we did was to take the genes that as they appear naturally, and we streamline with making them smaller, either with only 2 genes or with the whole set of genes. But in any case, the advantage of having light emission is that you have a direct measurement, a direct visualization of the activity of a given promoter in front of you. There are some disadvantages because the reaction for production of light is subject to some physiological controls, but in general, it's so nice to see bacteria emitting light that is great. And I'm not surprised that in many cases people use this reporter because of the kind of a nice signal that you get under given circumstances. But then, like the king or the queen of the last 10 years or 20 years in the area of reporters is GFP. So GFP, as you know, comes from these jellyfish that are found in many seas. There are many variants of GFP. Some of them are natural, others are engineered and so on, and it has a number of characteristics that make it to be very, very appealing. And what is also nice is that because of different mutations that different laboratories have been introduced in the original GFP, then there's now a whole palette of GFP variants, of fluorescent protein variants, in such a way that now you can mark the same bacterium with different colors, and each color will give you a different, will be an indication of the activity of different promoters. Once more, what we have done has been to make SEVA vectors with a whole collection of different colors that you have here, green, blue, red, yellow, etc. And then what you have at the end is this, this is something that comes from my laboratory. You have a collection of bacteria here, each of them with a different color. Isn't it nice? It's great, right? So it's visually just amazing. So, I don't know whether you have that, but once in a while we have kids from schools and so on that come to the laboratory to, see how a laboratory looks like. And this is one of the favorites, they get really excited when they see bacteria with different colors and so on. You tell them that this is a gene that comes from a jellyfish and so on. They get completely fascinated. Okay, so that's something very nice. Okay, so this goes for plasmids. Yes. Before this one? Yeah, okay. Okay. Thank you for the question. And please, every time I say a word or something that you don't understand, do what your colleague has said. Say, I don't understand that, please tell me, because that's the way to make progress. Okay, so a multiple cloning site is a small segment of DNA that has been engineered to contain many, many, many restriction sites, okay? And many of these restriction sites are typical restriction sites that are used for DNA cloning. So the idea, and thank you for the question, is that we have eliminated, these are frequent restriction sites, but we have eliminated them in the rest of the plasmid, and the only place in the whole plasmid where you have, say, sites available for cloning is only in a particular part of the plasmid. That is, because of that, it's called multiple cloning sites, because you have many possibilities of cloning DNA segments that are flanked by sequences that are amenable to restriction with these restriction enzymes. Am I clear? Okay, very good, thank you. I love it. Okay, good. Okay, so everything I told you so far is about plasmids. Now, as I anticipated at the beginning, you have a choice of putting your favorite DNA segment either in a plasmid or in a transposon. And the transposon has the advantage that we will shot that DNA in the genome of your bacterium, and therefore you don't need to care any longer about maintaining the plasmid with antibiotic resistance or anything like that. It will go integrated into the genome and it will remain there in general without any other further modification. Okay, let me spend a couple of minutes going through what is a transposon. Okay, so the origin of transposons is something that is called insertion sequences. So you know that in the genomes, once in a while, there are some sequences that happen to encode an enzyme that is called a transposase that recognizes the ends of these segments, and then it enables them to jump from one place to the other, okay? This is the typical case of what is called shellfish DNA. So this DNA here doesn't do anything. It does not encode anything of interest. The only thing that it does is to jump from one place to the other, period. It's the only objective in its life, just to go from one place to the other. But then occasionally it happens that two insertion sequences might be flanking something in between, okay? And then what happens is that the so-called transposase that is encoded by one of them recognizes not the end of the same insertion sequence, but the end of the other. And therefore, the entire segment can jump from one place to the other. And what you have in this case is what is called a transposon, okay? So a transposon is always formed by a kind of intervening DNA that is flanked by insertion sequences. So what happens when an insertion sequence jumps versus what happens when a transposon jumps? Well, if a transposase, if an insertion sequence gets inserted somewhere else, the only thing that can happen is that the target, the gene where it hits, will be mutated. I mean, there's no other possibility. In some cases, apart of that, sometimes inside the insertion sequence, there are promoters that go in one direction. So because, you know, the transposase has to be transcribed, and therefore inside the insertion sequence, there's a promoter. And therefore, if this element jumps somewhere into the chromosome, then the genes that are downstream of the insertion will be transcribed from the promoter that is encoded by the insertion sequence, okay? And what is the advantage of that? Well, that sometimes what you may call cryptic genes, genes that are silent, that never get expressed, when they have an insertion sequence that jumps in front of them with a promoter, it starts being transcribed, okay? That is what may happen with insertion sequences. But then when you have a transposon jumping from one place to the other, the transposon not only will interrupt a given DNA sequence, will also carry with it whatever phenotype is encoded by the intervening DNA that you have between one insertion sequence and the other, okay? So the kind of sum of what I told you is that both insertion sequences and transposons are mobile elements, they can move around, but the consequences of the insertions are a bit different because of these circumstances that I told you. So, as I said, when you have this insertion and you have a promoter nearby, it may, I mean, inside the insertion sequence, it may happen that this gene X can be activated because of the proximity of what you had before, okay? Or alternatively, you may have just an insertion in the middle of a protein, and then the gene is interrupted and nothing is made. Okay, so there are many, many types of transposons. One that is more useful for genetic engineering, well, there are two types that are more useful. I will discuss just only one of them. Is the so-called the transposon TM5. TM5 transposon. What is the TM5 transposon? Okay, so this is a DNA segment that is a mobile element, can move from one place to the other. And what you have here is a number of genes that encode resistance to three antibiotics: kanamycin, bleomycin, and streptomycin. And these segments, this segment of DNA with these resistances to antibiotics, is flanked by two insertion sequences, the one to the left and the one to the right. And in principle, these two should be very similar, but it happens, and this is part of the entire kind of trick of the transposon, and in fact, what makes this to be a good transposon is that one of the two insertion sequences carry a mutation that makes this insertion sequence inactive. However, it will still keep the terminal sequences that are at the end of the transposon that can be recognized by the good transposase that is in the other insertion sequence, in such a way that now this whole segment of DNA can move from one place to the other, okay? So there is some kind of geometry here. So the transposase acts on the ends, either O or I in correct orientation. Then when you have an insertion, you don't only kill or insert the corresponding DNA, but also you bring together with that insertion resistance to kanamycin, bleomycin, and streptomycin. And well, there's a cut and paste conservative transposition. So that means that if this transposon goes from one place to the other, then the segment from here to here will be reconstructed, okay? Also important is that the transposition is random. So it can go anywhere in the chromosome, and it is a good, say, property that can be exploited to in many cases. And also what is important is that the spectrum of possible insertions goes in basically any type of biological DNA. So the TNF transposition will work in E. coli, will work in bacillus, will work in yeasts, will work in mammalian cells. So once you have this element here, the system will work very well, because the only thing that is required for transposition is to have a segment of DNA that is flanked by these terminal sequences and the activity of the transposase that is encoded by one of the two insertion sequences, okay? As a matter of fact, you can even buy kits where you have a DNA segment flanked by the terminal small, say, motifs at the end of the transposition, and then the purified transposase. And that will be sufficient, even in vitro, to have this, segment to jump from one place to the other in the DNA. Okay, so this is the standard, this is the naturally occurring TM5. But can we, again, do it better than nature for our purposes? So let's see what can be done. Now, facts. The transposase, as I mentioned a moment ago, acts on the outermost 19 base pairs I and O ends of the transposition, okay? So that means that anything that is between an I end and an O end is in principle amenable to the transposition reaction. The other important aspect is that the transposase will act in cis, that means adjacent to the DNA that will act on, even if it is present outside the transposition, okay? And this is very important. So that means that in the naturally occurring transposition, you have the transposase inside the transposition. But as we are smart, we say, well, what happens if we put the transposase outside the transposition? Will it still work? The answer is yes. So even if you take the transposase outside the transposition, it will still be able to promote transposition of the segment of DNA that is flanked between the I end and the O end. So what is the consequence of that? That years ago, we started with the TM5 transposition, and then we developed what is called a mini transposition vector, with the idea of converting this naturally occurring element that is a TM5 into a tool that is useful for serious genetic engineering of different types of bacteria. So in this case, what we made was to construct in a plasmid a segment of DNA where we took, like reference, the I end of TM5 and the O end of TM5, and in between we had a marker, a selection marker, and then we engineer a multiple cloning site where you can put any gene X of interest. But then what we did was to put in cis, but outside this part of the DNA, the gene encoding the transposase. So what are, how this segment can be delivered to a recipient? Now you have to make an effort of abstraction to understand that, because it's not trivial. I told you a few minutes ago that there are some plasmids that can replicate in specialized hosts if the host produces from the chromosome this protein for replication called the peer protein. So you have a strain of E. coli, for instance, that is engineered to produce this protein from the chromosome. Then these plasmids that have an R6K origin of replication can replicate happily there. But if these plasmids go to another place without the R6K protein, it will make it inside the cells, but it will not replicate. Now, how can you use these, say, biological properties for delivery of mini transpositions into a cell? Well, what we did, or can be done, is that you construct a plasmid where you have an ORI R6K, that means conditional replication. It has an ORI-T, that means that it can be mobilized conjugatively to a recipient, and you have there an antibiotic resistance outside the transposition, And then you have this construct, you have this mini transposon here that has a selection marker where you have put some clone DNA, and then outside of this segment of DNA, you have the gene for the transposase, okay? So in sum, what you have is a delivery vector that can be mobilized into a recipient that can replicate in the donor, because the donor has the possibility to replicate the plasmid with an R6K or replication, because it has the peer protein, but at the same time, it can be mobilized to another recipient. So here comes the kind of trick. So in the donor strain, you have E. coli, a specific strain of E. coli, that has in the chromosome, has been engineered to produce the peer protein, the P protein here that is required for plasmid replication, okay? So you have here the donor, and then the donor gets together with a possible recipient. It can be another E. coli, it can be Ralstonia, it can be Pseudomonas, it can be Ralstonia, you name it. It can be any type of biological recipient. So you put them together, and then as you have all the machinery that is required for conjugal transfer of this plasmid into the other recipient, then you have conjugation, and then this plasmid is passed from here to here. But look, it passes from here to here, but as the recipient does not have the possibility to replicate this plasmid, the plasmid will be here, but will not replicate. And during the time of transient residence of this plasmid in the recipient, there will be enough time for the transposition machinery to bring about the transposition of the mini transposon into the chromosome of the recipient bacterium. Okay? And over time, as this guy cannot replicate, it will be lost. And if you have a smart, if you are patient, then the only, say, clones that have of the recipient that have received the phenotype encoded by the transposon are the ones that have incorporated the transposon into the chromosome. And we can do that because as we have a mixture of the two strains here, we can plate this, we can select this mating mixture in a medium that will kill the donor and will select for something that is present in the recipient and also something that is present and encoded by the transposon. And in such a way, at the end, bingo, you have your transposon that was initially engineered in E. coli and everything that ends up in the chromosome of your recipient cells there, okay? Obviously, to do that, you need conjugation. And conjugation is this system through which different bacteria exchange DNA, and with a pilus or a breach or something. This is a picture, a historical picture that appears in many books. And what we have engineered is a trick to simplify conjugation that involves to have a helper strain that will also give in trans the activities necessary for conjugal transfer, okay? So that means that in practical terms, all you have to do is to put a recipient cell, a donor cell, and a helper. And the helper will give to the system the enzymes, the proteins that are necessary for mobilization of this construct where we have the mini transposon to end up in the recipient there. So at the end, what you have is that these two plasmids will enter into the recipient, but will not be maintained in the recipient. And therefore, the only way through which the recipient can acquire the phenotype encoded by the mini transposon is if the transposon has ended up in the chromosome of the recipient, okay? Well, these tripartite matings can be visualized. The cells get together, and well, the resolution is not fantastic, but you see that you form these trios, where you have, in this case, you have two E. colis. You have here the helper, the donor, and the recipient. And the recipient is a Pseudomonas cell. So Pseudomonas are bigger than E. coli, and therefore you can differentiate even visually who is who. And clearly you have here two E. coli, one on top of the other. This guy here is probably passing the plasmid to this one here, and the two plasmids here are passed to the third say recipient or to the third actor in this trio. and then now you select this mixture in a medium that counter selects E. coli and selects for Pseudomonas, and also selects for the phenotype that is encoded by the mini transposon, then at the end, you know that the segment of DNA has ended up into this bacterium here. So for instance, if the transposon carries, or the mini transposon carries a resistance to kanamycin, for instance, then if you plate this trio in a medium with citrate plus kanamycin, then you have a good selection, because citrate is a substrate that E. coli cannot use, but Pseudomonas can use. So on the other hand, Pseudomonas in principle does not have resistance to kanamycin. Therefore, if you plate this in a selective medium with kanamycin and citrate, you kill these two guys, and the only ones that survive are Pseudomonas putida cells that have acquired the transposon that was engineered before in one of the E. coli cells. Right? Okay, so what is the advantage of transposons as compared to other plasmids? Okay, well, the main advantage is stability, because once the transposon gets inserted, it remains there forever. And it's there forever because remember that the gene for the transposase is outside, and therefore it is not inherited. So it's a kind of clean insertion. It goes into the chromosome and it stays there. There's no way it can move out in any case. And therefore that's good for stability, because there are no rearrangements and the transposase is lost. Then the other advantage is that selection is not required. If you have a plasmid, then you can be always concerned that the plasmid might be lost because you don't add antibiotic selection pressure. However, you have the gene of interest in the chromosome, then it remains there forever and you don't have to care about antibiotics. And this is also important, for instance, for industrial applications, because antibiotics added to a culture medium can be a considerable host. Then you can also use the same transposon system to execute various rounds of transposition, because as the transposase is lost, then all you have to do is to change the selective marker of a new transposon to keep on doing transposition and transposition. And finally, what is nice about the system, as I said, is that TM5 is the most host-independent transposition system, and therefore it will work in E. coli, it will work in Pseudomonas, it will work in Bacillus, it will work in yeast, it will work everywhere, and therefore it's a super good tool to integrate things into the chromosome or your favorite, say, bag. So in the last few years, there has been another system that has been developed with very, very similar, say, purposes, and it's based in the so-called mariner transposase. That is an eukaryotic transposase that has also these characteristics. Basically, it's the same story that I told you for TM5, but the advantage of mariner is that it has less bias for insertion in different parts of the chromosome. But basically, it's the same story, and I will not repeat the whole thing with the mariner transposase. So, in this slide, I just summarize some of the things I told you. So the transposon can be inserted in different parts of the cell, and then you plate for selection of the marker that is in the transposon and counter selection of the donor strain. So at the end, you can really have various, say, insertions, and some of them will produce mutants, and some of the mutants might be of interest for you. For instance, in this case, it's one of the thousands or millions of possibilities. The user of this particular slide was interested in finding insertion mutants that were unable to grow in low iron medium. So, a collection of insertions, and then you check in one medium or the other, and then you can identify, even sometimes visually, the clones that respond to that particular phenotype. So transposons are a wonderful tool for doing these things. So one added value of mini transposons is that you can very easily detect where the insertions have taken place, because as the transposons have some defined sequences, then you can have a random amplification of the nearby sequences next to the transposon, and then you get a collection of say, segments that can be amplified and sent to sequence, and immediately you have, after some PCR cycles, I don't want to go through the details, you have an instant identification of where the transposon has gone. Let me just finish with this slide indicating that in the last, about 10 years ago or so, we standardize the transposon vectors to make them compatible with the SEVA vectors, okay? So that means that now you have an entire panoply of vectors that are compatible, interchangeable. You can take segments from one place to the other. You can put it in E. coli. You can put it in Pseudomonas. You can put it here and there. And the kind of trick is that because of this standardization of the different segments, you can easily move devices from vectors to other vectors and also from plasmid vectors to transposon vectors. And therefore, you have an entire, say, list of tools that you have for making all types of detailed genetic engineering. And this is something that is just fantastic. Okay, so I'm going to finish this before we go for the coffee break. Then I will just tell you that if you are curious, then you may want to click in these important web pages. One is about the SVOL standards that I mentioned before, indicating the graphic standards, the different nomenclature, the symbols and everything for representing your synthetic biology constructs with these graphic forms that can be understood by everyone in the field. Then it's also interesting that you take a look to what is going on in the collection of parts. You can see what are the most popular parts if you want to make genetic engineering. Promoters are very popular, reporters are very popular, but you also have terminators, you have metabolic genes, you have small RNAs, I mean, you name it, and you see also the degree of standardization that you have in each case. Then all of you, I certainly recommend you to go into the IGEM webpage, where you have the whole picture and everything. Well, just read it, but read it if you have the time and the interest, read it with some degree of criticism, because all these aspects that I discussed with you about context sensitivity, emergence, and these things, these are issues that we all know that are there, but they are hardly ever mentioned, because no one wants to reveal to the general public our kind of secrets. So the big secret of synthetic biology is that things don't work in many cases as we propose to the world that it will work. We say we can engineer this, we can engineer that, and we do that for the sake of, to some extent, propaganda. But it's a bit different when we say to the external world as compared to what we have in the laboratories. And finally, if you are curious, you can go to this webpage with the SEVA plasmids. I don't know how many of you work with bacterial projects, or how many of you will work in the future with bacterial projects, but believe me, I mean, this is an absolutely sensational resource of plasmids and tools for making all types of genetic manipulations and all types of genetic programming in bacteria, mostly in gram-negative bacteria, but also in gram-positives, and more recently also in yeasts. Okay, so this is a wonderful repository of interesting, say, tools for different applications. Okay, so we go for coffee, and then we go quickly through the papers that I propose you to tackle for tomorrow. But now let's have a coffee break. Okay.